All right. So after telling you that to build an optimal pathway, you need to identify a good promoter. You need to you need to identify good plasmid. You need to clone the genes. You need to secure expression through either an inducible promoter or through a riboswitch or any other method for heterogeneous expression. Then you have all these things in front of you, and then the time comes to put everything together and to secure that things, all the genes that you want to put together, work with the right stoichiometry. Okay, and this is one of the most typical problems when you are making metabolic engineering. You know that enzyme 1 will act on a given substrate, and then will give you 2, whatever. But then what happens is that sometimes the balances between the activities of the different enzymes might be different, and then you run into different problems, and cells eventually might not make it. You know? So one possibility that we like the trivial possibility, but it's the most trivial, but in many cases is not the most efficient, is that you clone the genes by separate, and then for each gene you engineer a different promoter and a different expression system. Okay, so as there are many, then we have promoter 1, promoter 2, promoter 3. This promoter can be inducible for arabinos, this one by tetracycline, this one by IPTG. Having a polycistronic operon is also an issue, because it happens, and this is an observation that is made in molecular biology for a long time, that when you have a long RNA, and at the end of the RNA you have a 3' end, there are in the cell RNases that will chop up the RNA starting from 3' will go in that direction. Okay, so what happens then is that the enzymes whose genes, the genes of which are being encoded by the end of the RNA, in general tend to be expressed less than the ones you have at the beginning. Okay, so because you have these RNases. So one possibility is to engineer another screw turn at the end of the RNA, an RNA structure, a DNA structure that will form an RNA that will form a shape that will protect the RNA from the action of the exonucleases. Okay, so going back to what I said, you want to make a polycistronic RNA, but then the polycistronic RNA will be chopped up at the end by a nuclease, and then to protect that to happen, then you can engineer at the end of the RNA a structure that protects from that. There are various structures, in fact, that are known that do that. Okay, various promoters with various features in such a way that at the end everything could be expressed more or less well. So, and one of the main elements for that was to remove sequences in the RNA that could also be the substrates of endonucleases. Okay, so that's another aspect of the project that is represented here. So they identified that there was a site in the RNA sequence that could be also cleaved by a nuclease, and therefore it was removed. And well, by using different, say, alternative sequences, they were able to produce something that could not be cleaved by an endonuclease. Okay, so you see that this is not a piece of cake. Okay, so if you want to put together, like intuitively, you think, well, I want to produce a compound. You just put gene the start. Okay, but then you have to deal with expression, with stability, with Scheindel-Garnos, with you have to put it in a plasmid or not in a plasmid, to use promoters or to use riboswitches. You have to make many choices, and in many cases, it's not possible to predict which of the choices you want to work better. So that's the typical engineering cycle of trial and testing, right? Okay, so once you have everything in hand, then you clone the genes, you put them together, and in many cases, in that particular case, they made experiments, and then they observed with some consternation that the efficiency of the plant genes in the construct was not very good. Okay, so it was in fact very, very ridiculous, like 0.5 nanograms per ml. That's nothing, and therefore there were some issues with some rare codons that were present in the gene that didn't work very well in E. coli. So another round of optimization dealing with optimizing the sequence of the genes at stake that came from plants. So at the end, they could form artemisinic acid. But after all these tribulations of promoters, plasmids, rubber instability, etc., they were able to finally produce these two amazingly optimized operants. One of them for the first part of the pathway, and then the other for the second part of the pathway. Okay, a Nobel Prize for the Chinese lady that discovered artemisinin. I don't know. Well, I don't know if you follow this story, that you know, but there was a Nobel Prize in chemistry just a few years ago to a Chinese team led by a lady, the name of whom I don't remember, that had a paper in an obscure journal describing for the first time the structure of artemisinin and documenting that the drug was effective for treatment of malaria. Okay, so once that one topic receives a Nobel Prize, then no one else working in that project will be eligible for that prize. Okay. Okay, so we have a little. So I just wanted to give you these details on the structure, on the structure, on the whole process for artemisinin, because I think it's an exemplary case, as I said, of everything that is required for making a good catalyst for producing something that is valuable. So the last part of today's lecture, what I'm going to tell you is another project in which we have been involved more directly. That is about making applying some metabolic engineering methods to Pseudomonas butida, not for production or something, but for degradation or something. Okay. So and in this case, the challenges involved, and you will see, will be a bit different, because the intention in this case was not to use a bacterium for production of something in a bioreactor, but for delivery into an environmental place with a contamination problem. Okay, so in this case, the first question we had to solve is whether Pseudomonas butida can be programmed for working under an anaerobic regime. So the reason for that, and the interest for that, is that Pseudomonas butida is a good chassis for bioremediation, and I think that I'm trying to convince you of that at different points in this meeting, in this workshop. And one of the typical scenarios for Pseudomonas butida to work is in soil, if you want to use it as an agent for bioremediation. And it happens often that the soil is contaminated with a pollutant, in this case petroleum, for instance, as well as petroleum, but and the petroleum can go deep into the soil column, but it is quite clear that the oxygen will be available only in the upper layer of the soil and will not go farther down. Okay, so if we want to have a bacterium able to work through the entire soil column with a bacterium that can deliver the activities of interest both under aerobic conditions, but also under anaerobic conditions, or at least conditions where you don't have out of oxygen. Okay, so Pseudomonas butida by default is strictly aerobic. Okay, so it needs oxygen to work, and if it does not have oxygen, it dies, period. And therefore, for this type of applications, it will not be a good chassis. However, we say, well, maybe we can change the metabolism first to make it work well under anaerobic conditions. So to do that, what we did was to go to a metabolic model, and there are various metabolic models of Pseudomonas butida. Metabolic models are more the business of systems biologists, so I will not get much into that. But as I said, now it is possible to have a very good picture, a very good representation of all reactions that happen in a given bacterium on the basis of the genome. Okay, so what we know from the metabolic pathways of Pseudomonas butida is that the system lacks completely genes for anaerobic respiration. So that means that the moment you don't have oxygen, no way cells can survive. There are some genes, but not all of them, that enable fermentation of some substrates, like lactate dehydrogenase, but it's not complete. And the consequence of that, in any case, is that the strain of P. butida that we use, that is called KT2440, cannot grow under anoxic conditions, period. I mean, it doesn't grow at all. Okay, how can we overcome the state of affairs? Well, there are two major reasons why one strain may not be able to grow in the absence of oxygen. So one of them is that the level of production of ATP may not be sufficient, but also that the redox balance of the metabolism may not be adequate. So in Pseudomonas putida, the way to produce ATP is through an alternative pathway of glycolysis that is called the Edner-Dodorov pathway that does not really produce a lot of ATP. It produces like half of the ATP that is produced through the standard glycolysis. So in the absence of oxidative phosphorylation, then ATP perhaps can be generated through substrate level phosphorylation to fulfill energy requirements instead of the typical glycolysis. But then it happens also that in the default metabolism of E. coli, then there's NADH, NAD, NADPH, NADP, and then it happens that under reductive conditions and in the absence of oxygen, then you accumulate a lot of the reduced forms of NAD and NADP, and therefore that can be a kind of poison. Cells may not grow any longer for that. Okay, so what we did was to take a look to different genes there, and then we found that P. has a partial acetate synthesis pathway from acetyl-CoA. Okay, so that's it has the enzyme here, even though it does not have this other enzyme that happens in E. coli. Okay, so that means that in this case, then we would need to add one more enzyme to have a complete activity able to produce the whole step here. Okay, so I have this root here, PTA, great, and we can monitor that the genes and the activities are there. Okay, so how can we complete the route that eventually will produce ATP at the level of phosphorylation of ADP, but not coming from other sources of active phosphorus, but from acetyl-CoA. Okay, so in other words, so we want to bring about this reaction, convert ADP into ATP. We want to produce ATP to compensate the deficit that we have before of ATP, and we have only this part of the pathway. We don't have this part of the pathway. So what we did was to bring this gene from E. coli. Okay, so we have this enzyme from E. coli, and then we put it in front of a promoter that can be induced and everything. And then once you complement this part of the pathway with this other pathway, with this other part of the pathway, then you can have this direct phosphorylation of ADP to ATP from acetyl phosphate. Okay, so and that's one thing that was important. Well, these are some data here that in fact the pathway, the complete pathway worked very well. So these two first steps were produced by Pseudomonas metabolism, and this other step could be brought about by introduction of the enzyme ICKA from E. coli in such a way that we could get this substrate level phosphorylation, and therefore we could get more ATP than normal. But there was the most difficult part to crack was not ATP production, but how to eliminate an excess of NADH. So in that case, the situation is that we have pyruvate, acetaldehyde, and then we know that bacteria that grow under anaerobic conditions can either respire anaerobically into or either nitrate or nitrite, or convert carbon substrate into several low molecular weight molecules. But you know what would happen in this case? Okay, in this case, what we need is to enter into the system a gene or a number of genes able to kind of eliminate as much as we have NADH, because NADH accumulation is one of the reasons why you don't have a good balance of redox, say, currency for allowing the viability of the cells. Okay, where can we get these enzymes from? Well, we went to another microbe called Pseudomonas mobilis, that is an obligate anaerobe that has a very, very good system for starting with pyruvate and, so to say, burn NADH into NAD, and at the end producing ethanol. So what we did was to again generate a small segment here, optimize expression, put it in Pseudomonas putida, and then monitor that indeed the enzymes were there and that enzymes were produced and were very well. Okay, so then finally, as we had a pathway for production of ATP and another pathway for oxidation on NADH, what we did was to make a hybrid pathway, a hybrid operon. When we have one promoter, I mean, this will be an operon, right? One promoter, Schneider-Garner gene 1, Schneider-Garner gene 2, Schneider-Garner gene 3. We didn't have to put a motif here to control mRNA stability, because what we had without doing that was good enough for our purposes, okay? Okay, so what we then checked was the possibility to, to see how this strain that was engineered work under anaerobic conditions. And then what we observe is that under non-treated or the natural strain will die very quickly under anaerobic conditions. When you enter into the cell, one of the features that I just mentioned, it survives better. When you add the two, then it survives quite well. Okay, so that means that you have this at least 1 or 2 orders of magnitude from here to here, and that means that the cells can really live well in anaerobic conditions, and as contrary to what we had with the natural strain that cannot do that. And then if you make another experiment of monitoring cell viability after some time, that is what you do with this compound here called propidium iodide. Propidium iodide will stain cells that are dead, okay? So if you are dead, you have a lot of propidium iodide capture. If you are not dead, then you have less propidium iodide capture, okay? So you see that when you have, when you don't have the pathways, then cells die. Most of the population die when they're exposed to the absence of oxygen. And when you do the contrary, I mean, when you put the pathways in this Pseudomonas putida, then cells are, I mean, it's not that they live very well, but at least you have a considerable improvement in the survival. Okay, and you can also monitor that cells are alive because you can put them a biosensor of vitality. For instance, you have this protein here that that gives some fluorescence when cells are have a good vitality, because this fluorescent protein requires some intracellular metabolites to give some fluorescence. So that means that when you have fluorescence, that means that you are well alive and kicking. When you don't have fluorescence, that means that you are in very bad shape. Okay, so you see here under aerobic conditions, the wild type, and the strain that is engineered, where you can see that cells emit fluorescence even if they are in a situation with very little oxygen. Okay, so then the situation here is that we have a starting bacterium that of ATP, and another pathway for eliminating excess of reductive power. Okay, and we see that this strain, when you put it in under anaerobic conditions, it survives well. Okay, so that's good enough. Okay, but that's not the end of the story, because we wanted to use that strain for actual bioremediation, and the compound that we wanted to bioremediate is this one here. It's a haloalkane that's called 1,3-dichloropropene. So this is a compound that is used in the agricultural sector, and also as one of these liquid cleaners that you have in dry cleaning. Okay, this is a compound that is very toxic and is very difficult to degrade because it has a carbon at the extremes of the chain that are blocked by chloride bonds, and this type of situations make the molecule quite difficult to chew up. Let's put it like that. Okay, so the idea was to develop a bacterium able to degrade this compound under anaerobic conditions to see whether at the end we could put it in soil and have it working. So there's a pathway for degradation of this compound that comes from another Pseudomonas, the Pseudomonas pavonaceae, but this pathway obviously works only under aerobic conditions because it comes from a strain that is a strict aerobe. So the idea was to take this pathway and again engineer a artificial operon with all the genes involved in the pathway, and then examining the different enzymes one by one, make sure that they work, optimizing expression with a promoter, with Schneider-Gartners, with all the different optimization necessities that you have to optimize expression of one of these constructs in your bacterium. And then, you could see that when you put this strain in a plate, I mean, the pathway worked very well because it has all the genes there. Now, the big question, does the system work under anaerobic conditions? Because that's at the end what we wanted. Okay, so the situation is this one. So when you have this bacterium that has been engineered with all these features and you put it to work under anoxic conditions, then what you observe is that the concentration of the initial compound comes down and the release of chloride goes up. So that indicates that the compound is being degraded actively, okay? And therefore that means that the strain that you have produced for this type of applications work very well. And work very well because neither Pipitida KT2440 or the Putida, the strain that produced the genes or had the genes for degradation of this compound, is able to degrade 1,3-dichloropropene under anoxic conditions. Okay, so it's a good indication that the metabolic engineering, say, endeavor was adequate. Okay. Okay, so that's okay. Okay, so look, in the case of artemisinin, the objective was to produce a compound. Okay, but you can do that in a bioreactor, and therefore the only thing that you have to be concerned is about having the pathway working very well. If you want to use the strain or make metabolic engineering for bioremediation, you have to think not only on the pathway, you also have to think in other environmental circumstances. And in the case of the chloropropene, the circumstance was that you wanted the pathway to work under anaerobic conditions. So that meant that you had to engineer not only the pathway proper, but also the pathway plus other characteristics in the strain that enable the strain to work under anaerobic conditions. Okay? But you can have still another issue, and is that how do you, in an environmental situation, in what physical format do you deliver your activities to the target site? You want to, you have a kind of planktonic suspension of the cells, and then you just put them around, or you want the strains to stick to the surfaces that are coated or are contaminated by the compound. You have to think also, and this is part of the metabolic engineering agenda, in the physical format that you want to give the catalyst for optimizing the activity that you want to put into the environment. So we enter into this other area of synthetic biology connected to metabolic engineering, that is synthetic morphology. Okay, so you think not only on the pathway, not only on the environment, but also in the physical shape, in the material shape that the catalyst must generate, must acquire to have an optimal activity in the conditions that you want. So the degradation of the target compound, in this case, is another chlorinated compound that is called chlorobutane, but similar to the one that we had before. The issue was not only to engineer the pathway, was not only to engineer whether or not it was working under anaerobic conditions, it was also about working or engineering its lifestyle, okay, in environmental conditions. So, therefore, there are various issues that we have to tackle. Can we program Pseudomonas putida to switch between planktonic and biofilm lifestyles? Does catalytic efficiency change with such lifestyles? Well, we'll see what happens. So this slide is similar to one I showed you a few days ago, indicating that there's this intracellular small molecule that is called cyclic DGMP, that has different levels, and when the intracellular levels of this compound go up, then cells tend to stick to surfaces. When they go down, they tend to be planktonic, namely to hang around in a liquid medium, okay? So this is important because if we had a way to control intracellular levels of this small molecule, then we can bring about either biofilm formation or complete dissolution of complete suspension of the bacterial community in the liquid, okay? So you may remember in one of the previous talks, we talked about something similar in which this was manipulated through illumination with light, okay? But in this case, we are going, as you will see in a moment, to do something similar, but the control will not be by light, but will be by a chemical inducer. But the bottom line, in any case, is that the engineering aspect is that a part of the pathway, we had to engineer adhesion or not adhesion to a target surface, depending on the exposure to a given inducer. Okay, so this is something that just to refresh your memory, because we mentioned that before. So the intracellular levels of this molecule depends on the interplay of two activities. One of them is an activity that gives cyclase activity, and the other is other enzymes, other set of biochemical, activities that are phosphodiesterases. So you have a lot of cyclases, then you have a lot of decycled DMP, and therefore you have a lot of biofilm, and you have a phosphodiesterase. You destroy the intermediate molecule, and therefore you dissolve the biofilms, and therefore the cells go into solution, okay? Okay, so in this case, instead of having a system, an optogenetic system for optimization of the expression, we use yet another expression system based on a transcriptional factor called CHNR that responds to this compound here, that is cyclohexanone. So cyclohexanone is an inducer that is not recognized by the metabolic network of Pseudomonas putida, and in that respect, we can say this is an orthogonal inducer because it induces the transcriptional factor here without interacting with anything else. So just to see how this thing worked, then we made some experiments with GFP, and then we observed that the induction was very nice, so it was great, so no problem with that. The difference between non-induced and induced conditions was very good. There was very little noise, and the induction was very good. So we made the choice of using this expression system for our purposes. So in this case, in one instance, what we did was to express conditionally the genes for formation of biofilm under the control of this cyclohexanone-dependent inducible system. And then this gene that is called GADQ is a cyclase that will cyclate GMP and produce this intermediate. And therefore, the prediction is when you have more of this enzyme, you have more of the intermediate compound, and therefore you have more of the biofilm. And this is exactly what you observe. Okay, so depending on the concentration of inducers that you add in the medium, then you have more or less biofilm form. There are various ways to monitor formation of biofilms. I didn't have the time to give you a class on biofilms, but basically what the red bars indicate is a higher or lower degree of adhesion of the cells to a surface. Okay, so that's something that you can see there. Okay, so you can in fact monitor that visually with fluorescence microscopy, with confocal microscopy. And then what you observe here is that when you induce, when you don't have the system induced, then cells barely stick to surfaces. When you have the system induced and you produce the cyclic GMP, then cells are, you can observe them stuck to given surfaces, okay? So by the same token, if you put under the control of cyclohexanone a gene that encodes a phosphodiesterase that destroys the small molecule that brings about formation of biofilm, then what you observe is that the titles of the biofilm goes down. Okay, so these are the controls. The biofilms of the controls, the blue ones, remain more or less stable over time and over different concentrations of cyclohexanone, but the strains that have this construct, when you expose them to cyclohexanone, you see that the level of biofilm formulation comes way down. Okay. Okay, so in other words, you have this system of controlling biofilms or not controlling biofilms. Now, you put together, again, everything the same strain, and then on the one hand, you generate in the laboratory an E. coli strain that has a complete biodegradation pathway for this chlorobutane, Again, that has to be optimized with a good promoter, a good Chandler-Garno, gene 1, Chandler-Garno, gene 2, etc. But at the end, you have a route for biodegradation of 1-chlorobutane. And then in the same bacterium, you have also engineered the possibility to make cells sticking to surfaces and forming biofilms. Okay. So what you have here is the situation that, the efficacy of the catabolic pathway, depending or not, or whether the cells are stuck or not to a surface. Okay. So when you have the cells in a planktonic state, you have a level of activity. When you have the cells growing, instead of glucose, you have an succinate, you have low. But then if you trigger formation of biofilms, you see that the activity of the degradation goes way up. Yeah? And the reason why it goes way up is because when you have a community where all cells are together, then the catalytic efficiency is way more intense, simply because in many cases, some of the intermediates of the degradation go out, and then if they go out and they don't find any neighbor, the compound will be diluted. But if the compound that goes out finds another neighbor, it will be channeled successively through various strains, and at the end, the catalytic efficiency will be much, much better. Okay. So, in sum, yeah, you can make metabolic engineering at various levels. The simplest, the ones that involve just one thing is the optimization of a pathway. And this is what we have seen with artemisinin. And artemisinin is one of the cases where there are many, many difficulties that you have to solve. In other cases, fortunately, you don't have to solve so many problems, but it still is not easy. But in the case of artemisinin, all it was about is to make a strain with a perfect pathway, period. For your applications, you need not only a perfect pathway, but you need also to engineer some physiological traits, because the strain that you engineer is going to perform, or will be performing, or you expect it to be performing in a scenario in which you require other physiological traits, a part of the pathway, to give a good activity and to do the job that you have engineered the strain to do. But third, a part of that, you may want to engineer some type of morphology in the catalyst, not only the pathway, not only the physiological conditions, but also the physical format that you have for delivering the catalytic, say, activity. And this is what I show you in this last case, where a part of the pathway and a part of other things, we engineer also this formation of biofilms or not. That is not related directly to the metabolic activity, but it helps, as you can see there, to produce at the end a good level of bioremediation. Okay, so this idea of synthetic morphology has kind of boomed in the last few years, because it is clear that depending on the physical shape that you have in a bacterium, then things may be better or worse. So, for instance, one very popular way in the last few years to play with the morphology is to engineer cells that, instead of being like a rod, are round. You know? So a bacterium is like this. Okay, but this shape is not casual. It is a result of a genetic, say, wiring of some of the genes of this bacterium, and you can make some mutants that, instead of having a rod, will form a ball. Okay, and then when you have a ball, then you optimize the exposure of the catalyst to the external medium. And so, another possibility, and this is something that is enabled by some of the tools that I have explained to you in these last few days, is to make, for instance, a consortium where you stick cells to each other, not to a surface, but to stick cells to each other, and then you form like clumps or clusters. And then these catalytic flux are more efficient than having just the cells separated one from the other. And well, this is a kind of endeavor that is you have to bring into the entire list of considerations for engineering that at the end will produce, or are expected to produce, catalysts that work well. Okay, so I think that we are done with this story. Yeah, because this is something I give you a aperitif for tomorrow. And well, we will discuss that tomorrow together with other issues, how to go from working from a small level all the way to a planetary level, but that will be for tomorrow. Okay, so ladies and gentlemen, I think that this is basically the 2 hours that I wanted to tell you about metabolic engineering. I don't know whether you have any question. Have you worked with metabolic models in your work? No? No one has worked with metabolic models? Okay. Okay, well, sooner or later, you will work in your future profession with bioengineering, and so I think that you will go through metabolic models, but at least I wanted to give you a kind of flavor of what metabolic models are for and what are they useful and all this. Okay, any other question, any other comments on metabolic engineering? No? Okay, great. So we'll break for coffee and then we'll return to discuss a little bit the planning for the IM things, and also to hear our friend to give his seminar or his little presentation on the paper that he volunteered to discuss, okay? Okay, great. Yes. You are there? You sent, okay, what happens is the following. Many of the emails that you send to me for unknown reason go to the spam, and then I don't see them. And then, okay, but thanks that you told me, because I go to the spam maybe, and for sure I will find it, okay? Okay, great. Yes. Any other question? Any other comment? No? Okay, so let's go for coffee and then we'll return.